This clip is going to give you the background information you need to know for the genetics wrap pertaining to protein synthesis. Protein synthesis is something we're going to do right after genetics because essentially what genetics is are genes that are duking it out, right? Those two alleles. And when we say that a child ends up with brown hair versus blonde hair, what we're talking about is either protein synthesis of the pigment or not, in which case would be blonde or lack of pigment. So these examples that we talk about in class directly relate to proteins. In order to understand proteins, you can look at this figure from your book. This figure gives you an overview of protein synthesis. What I recommend is going back and getting your endomembrane system protein synthesis handout out. I believe it's labeled as Protein Synthesis Help. And we did it in class together right before the midterm. And we're just going to add some detail to that in class. For instance, we're going to give the creation of RNA in the nucleus, we're going to give that a name. That's called transcription. So the copy of the DNA into RNA is called transcription. That's a process, and that happens in the nucleus. Now that RNA leaves and hooks up with the ribosome, is fed through in a process called translation. So you could add that to your protein synthesis help handout. And there you can see the polypeptide chain coming out. That, of course, is not a protein yet. It is just a polypeptide chain. You know at that point it has to go into the endoplasmic reticulum to get folded, and then it shoots over to the Golgi apparatus to be stored maybe for the short term, certainly flagged, and sent out to do its duty. So we're going to look at these processes in a bit more detail, and in order to understand them, we really need to understand RNA. You've had a bit about DNA. You know that the base pairing rules are A to T, G to C. You know that DNA is housed in the nucleus. You know that in eukaryotic cells, DNA is conserved. It needs to be protected, and so it never leaves. Therefore, when we're making proteins, which we're doing all the time, we have to make a copy of it. But that copy is more of a cousin versus a twin sister. Okay, so RNA does encrypt the same message, but it looks a little bit different. So let's compare DNA to RNA. First, DNA is double-stranded, and RNA is only single-stranded. So you notice there's nothing else over here. It's just a single strand. The other difference is that you see these U's, and this is uracil, so it does use a slightly different language than DNA does. Although the message is the same, the order of amino acids, that message is the same. It just looks a little different. So it's like saying hello in English and Spanish. The message is the same, but it sounds a little bit different. So you can see over here, A to T, G to C, we're going to have different base pairing rules. We're going to go A to U, G to C. So when you are going through transcription and you're moving from DNA to RNA, these are the rules you're going to use. Let's do an example. So if I have DNA strand that looks like this, A, T, G, C, if I asked you what the RNA strand would be that's made, you would tell me U, A to U, A, that T is read as a U, C, and G. And so from DNA, RNA is made. Now you can go RNA to RNA too. Let's maybe label this DNA and RNA. Okay, so let's go RNA to RNA, which we'll do a little bit of in class. So let's do that U, A, C, G. RNA to RNA base pairing rules is A to U, G to C. So it's A, U, G, C. Okay, so we'll get a chance to practice with that a little bit in class, but you should know that general base pairing rule from DNA to RNA. So in class, what we're going to look at is the process of transcription and translation. I'm going to have everybody pull out that protein synthesis help handout, and we're gonna go through that a bit more.